And it's a real pleasure to introduce Frank Szoka. Frank and I have known each other for how many years is it now? It's getting on for 40 or so, maybe a little bit more. And you know, I just want to emphasize a couple of things. That Frank has been a dominant figure in the area of lipid nanoparticles, liposomes, and drug delivery for all of that time, and has really played a formative role in some of the efforts that I've been involved in. You know, particularly in the formation of liposomal or lipid or liposomal systems for drug delivery. Pioneered the procedures such as reverse phase evaporation for getting very large encapsulated volumes, extrusion. Came up with some of the first ways that we could actually make these vesicles in a reproducible, scalable way. Mechanism of encapsulation of whatever, whether it's small molecule drugs or larger proteins and oligonucleotides, and and also mechanism, the biophysics really of the mechanism of intracellular delivery of oligonucleotides in complexes. So it had a profound influence on the field and continues to do so. So it's really you know, there's very few people that can say as a result of their career that they changed their therapeutics and they've changed science, but Frank is one of them, and it's a great pleasure to introduce him. And I got to say, 6 decades in the cancer nanomedicines or whatever. This is getting a little daunting, Frank. We have known each other for too long now, but let's hope this continues for another 6 decades. Anyway, Frank, take it away.
Thank you very much, Peter, for that kind introduction. I am delighted to be here and delighted to be speaking to this group, and it gives me a chance to look back on 6 decades. So it's a pleasure to be here. So I'd like to talk a little bit about history that Peter was just referring to. And it all started with Bangham back in 1964. And there's a nice little article that discusses the early history that I've noted on the lower left. And the names that come up often are Demetrius Papahangiopoulos, who was my mentor and friend, and we were co-founders of Sequus Pharmaceuticals. Jerry Weissman, Gregory Gregoriadis, Brenda Ryman, Walt Shaw, and then from the biophysics side and biochemistry side, there were the giants, Van Dienen, Tommy Thompson, Dave Deemer. And the ones that I'd like to call out were Demetrius, who started one of the liposome companies and was a big contributor to the biophysics of liposomes. Gregory Gregoriadis, who really was, I would say, the shepherd of the liposome group. He started the first liposome drug delivery conference, the Gordon Conference on Drug Carriers. He started the liposome NATO meetings. He's run the London liposome series for 30 years, and he continues to be very active in the liposome research days. And the other person that had a huge influence on liposome technology was Walt Shaw of Avanti Polar Lipids, who made high purity lipids available to us all to do various imaginative things. So those sort of what I would call the giants of the early liposome field. And as we go forward to the next slide, that's led us to what I would call a versatile drug delivery nanotechnology. So liposomes, first and foremost, are based upon an extensive understanding of the biophysics. There's superb GMP lipid sources from around the world, Japan, Europe, the United States. There's robust manufacturing protocols, extrusion, now microfluidic systems. There's highly developed analytical protocols. There's a very good understanding of the PK and biodistribution of liposomes in humans. And a huge advantage of liposomes is that they're biocompatible and biodegradable. So they are made out of materials that make up our cell membranes. The regulatory pathway is defined by the FDA and the European regulatory authorities and is well understood. There's new advances in patient staging for the EPR effect are becoming a reality, and multiple drugs delivered in the same liposome is now a promising commercialized product. No other system can really claim that, and that puts us in the area that liposomes really are at the top of the nano systems, and top not only in commercialized products, but also in the size range, because they run between 60 and 140, 50 nanometers in size. And so that would be just at the edge of what I would consider a nano system. Could I have the next slide, please? Because of this, many liposome products have been commercialized, the first one being Doxyl in 1995, followed by Ambisomes, and then Abelset, and then by a number of others, Marquibo, Onnavide, and most recently Viexios, which is 2 drugs in 1 liposome. So liposome products have successfully entered the market, but to be perfectly honest, as Pieter had pointed out just a few minutes ago, they're a very small segment of the market, maybe $2 billion worth. And the new lipid nanoparticles, they're selling, I don't know, 20 billion, $30 billion worth of vaccines. And antibodies, of course, are the kings of what I would call cancer drug therapies. So liposomes are important, but on the lower end of the commercializable scale. Could I have the next slide, please? But the thing that I'd like to point out is that liposomes as cancer nanomedicine arose from startup companies. So Sequus, founded by Dimitri and myself, led to Hermes, led to the Taiwan Liposome Company, led to Merrimack, now led to Zone 1 Pharma. On the East Coast in Princeton, the Liposome Company was started by Jerry Weissman, and the Liposome Company acquired a large number of liposome experts, chief among them Peter Cullis. There was Rudy Giuliano, and that led to Pieter starting a liposome device company, Northern Lipids, and led to Inex, Protiva, and many others in there, and also Celator. And so Pieter and the group at the BC Cancer Center turned out in the upper echelon of these company foundings. In Southern California, Balderschweiler from Caltech started Vestar, which became Nexstar, which became Molecular Express, and now this is all owned by Gilead. And then there's many others, and I haven't put them all up here, but one is a small San Diego company that's selling a liposome encapsulated buprenorphine. So all of these cancer nanomedicines came out of small companies. Some of them became large companies. Next slide, please. I just want to turn to what I call the poster child of liposomal drug delivery. As Terry Allen has called it, Doxyl was approved in 1995 for Kaposi sarcoma, subsequently for ovarian carcinoma, and later for multiple myeloma. As a liposome, it really is a very tight liposome, and it stabilizes drug from release in the bloodstream. It has a very prolonged circulation, and it has passive targeting to tumors via extravasation through gaps in blood vessels. Next slide, please. Some of the technology that made Doxyl possible was a pegylated lipid that is embedded in the liposome surface. It's part of the composition. It provides a polymer coating that extends about 5 nanometers from the bilayer. And on the inside is a nanoprecipitate of doxorubicin with ammonium sulfate. And you can see this on the right-hand side. There's a cryo-EM, and Doxyl is noted for having what's been called the coffee bean shape. And the drug is encapsulated on the inside. And if you look at the PK of this formulation, it has a very long circulatory half-time. It's 2.5 days. And you can reach into a patient and take out a blood sample 30 days later, and you can measure liposome encapsulated doxorubicin in the patient. So it provides a very slow release of doxorubicin to tumors. Could I have the next slide, please? So what were its design features? It has about 10,000 doxorubicin molecules per liposomes, long circulation, 100 nanometer diameter. It's very stable. And because of this, it was very easy to prepare filter sterilized preparations. It's one of these products that's stored as a liquid, but it can also be frozen. And it has been on the market now for almost 28 years. Can I have the next slide, please? And fortunately for the advance of this liposome, it was first tested in Kaposi sarcoma patients. And these are patients that have a disease that was associated with HIV, and it involves an infection of endothelial cells and their proliferation and breakdown of the endothelial lining of the blood vessels. And when the liposome is injected, in this case, the pegylated liposome had indium in it. And so this is gamma scintigraphy. And you can see where the liposomes have distributed. Notice in the eye, these are the blood vessels. You have the liver and the spleen that are well outlined. So liposomes are taken up in the liver and spleen. Notice in the bone marrow, in the spinal cord, as you move to longer periods in time after injection. And you can see a fabulous extravasation of the liposomal drug into the tumor. And so the lesions on the patient really were the targets for the liposome. And liposome dox was curative for Kaposi's sarcoma patients. And when the drug approval documents were submitted to the FDA, there were only 126 patients treated. Of these patients, quite a few of them passed away from AIDS. And so they really couldn't be evaluated for the performance efficiency of the doxorubicin liposome. But there was a case physician who reviewed all these cases, and he went through and pulled out the 25 or 26 patients who survived and their tumors were eliminated. And based upon those 25 or 26 patients, Doxyl was approved for treating KS patients. And at about the same time, fortunately, an antiretroviral drug came on the market. And so the incidence of KS has dropped back to a very low level. But this sort of set the stage for the idea that liposomes can extravasate into tumors. Can I have the next slide, please? And this is the cartoon for the EPR effect that was promoted by Mayata in the 80s, that there would be openings or fenestrations in the endothelial cells in the tumor where material could leak out and percolate through the tumor and interact with tumor cells. Whereas in healthy tissues, there's a well-defined and barrier with tight junctions, and it's harder for material, particularly large material, to get out unless they have some sort of active mechanism like macrophages or T cells. So this was what was believed to be working in KS patients and was hoped to work in other types of tumors. Could I have the next slide, please? Now, it turned out that Nexstar or Vestar had introduced a liposome encapsulated DTPA called Vescan. And that was a diagnostic product that was on the market for a short period of time. And that was used in a large number of different tumor types to explore for the existence of the EPR effect. And so this is just an example of the tumors that it was looked at in. And the result of that was maybe 50% to 3/4 of the tumors exhibited an EPR effect of various amounts. So this, again, gave hope to the liposome community that liposome encapsulated drugs could be useful in these tumors if they could be identified. Could I have the next slide, please? And so Doxyl became a very widely used drug based upon the early evidence of EPR in human cancers. And it has a phenomenal safety protocol. One of the principal limitations of doxorubicin is it causes a cardiomyopathy. And when it was given in Doxyl, the incidence of cardiomyopathy went way down. And some patients actually have been treated over the years by up to 10 grams of doxorubicin as the Doxyl product. And so it has shown good efficiency in ovarian carcinoma. Patients that are on this often can have a 3, 4, or 5 year extension of lifespan until finally the tumor escapes from the drug. But there's still a big gap in efficacy. So with the liposome, you can deliver a much higher dose for a longer period of time, but there are limitations. The EPR, as I've already indicated, isn't particularly strong in all tumors, particularly metastatic tumors. The liposomes interact with the MPS system, the macrophage system, so it often blunts the magnitude of the therapeutic advantage by killing macrophages. We made the liposome Doxyl very tight, so it's suboptimal for drug release rate from liposomes into the tumor. And then optimism was based upon dose effects in mice species that really don't cross well into human species. And then we also have the problem of poor clinical study design, which that comes about because often pharmacologists aren't familiar with the properties of sustained release of liposomes. So they're either dosed too frequently or not frequently enough. Next slide, please. The Merrimack as a company introduced a couple of very useful technologies for stratifying patients for either good or poor candidates for liposomal drugs. And so they imaged the EPR effect in pancreas tumor patients using an MRI signal. That is, they would put in an iron particle, and then they would see if the tumor could be observed in the scanner. And they found that there was a correlation between the ability of the iron particle to go to the tumor and the response to their irinotecan product in patients. And they published that in 2017. So it's very possible to stage patients. And on the next slide, there's you can use PET imaging. And in this case, copper PET was used to look at in the enhanced permeability effect in treatment responses in metastatic breast cancer. Again, this is something that came out of Merrimack. So now it's possible when you take a liposomal drug product to the market to stratify patients based upon the ability of a surrogate marker to identify which patients would be better candidates for the treatment. Could I have the next slide? Okay, so that's the background. This is what I've told you so far. Liposomes can provide an extended circulation. In certain cases, they can penetrate tumors. And then there's a need for targeting and payload release. And all of that is a function of delivery. And what I'd like to do now is look at some of the work coming out of our lab recently in these three areas. Next slide, please. The first is long circulation advantages of polyethylene glycol. And this is work that was done by Paul Kerstead, and it involves the synthesis and preparation of 5 additional polymers attached to lipid anchors to look at their ability to provide long circulation. So this is the cartoon on the lower left. The polymer is embedded by a lipid anchor into the membrane. It has a structure, either a mushroom or an extended form, away from the surface. Could I have the next slide, please? But one of the disadvantages here is that the PEG has a high viscosity, which isn't ideal for clinical use. Its intrinsic viscosity is quite large. Some patients have pre-existing antibodies to PEG, and depending upon who's done the survey, between 2 and 25% of people nowadays have pre-existing IgM antibodies. And what that leads to is when the liposome is injected, depending upon the dose, if it's a small dose, the liposome can be eliminated very quickly. The other disadvantage is that it became to be known that there was an accelerated blood clearance phenomena, that patient might be injected the first time with a liposome modified by PEG, but when it was injected the second time, between 2 to 30 days later, there was a rapid blood clearance and elimination in the liver. And so the question is, why does this come about, and can it be avoided? Can I have the next slide? And here's an example. This effect of the accelerated blood clearance was described by Dams out of the Gert Storm laboratory back in 2000. The first dose gives you a very nice elimination curve with a half-life of about 24 hours. The second dose, 5 days later, is immediately eliminated, and the liposomes are taken up by the liver and spleen. And so the mechanism was proposed was that the first dose binds B cell surface receptors and triggers production of an anti-PEG IgM. And that leads to this rapid clearance on the second dose. So you can overcome this by giving very large doses of the pegylated liposome, and you can clear out the IgM, or in this case, we're looking for other polymers that might exist that could avoid this. Could I have the next slide? And Paul synthesized 5 different polymers, hydroxypropyl methacrylamide, PVP polymers, PMOX, or an oxazoline polymer, a morpholine polymer, and a dimethyl acrylamide polymer. All of these are water-soluble polymers. They can be made in very, very tight size ranges, and they could be linked to lipid anchors to insert into a liposome. Could I have the next slide? And here are the properties of the polymers. These are the structures, and this is the molecular weight. He synthesized high molecular or medium molecular weight polymers with very narrow polydispersities and he measured the viscosity. And so for PMOX, it has a reasonably high viscosity of 2.01, and PEG is down here of 0.234, whereas all of the other polymers have lower intrinsic viscosities. These on the next slide were embedded into liposomes, a Doxyl-like formulation with cholesterol, hydrogenated soy PC, and a fluorophore di-ID. They were prepared by high pressure extrusion. And with that, they had a very tight molecular weight range from 2 to 3. And liposomes were formed all with a diameter ranging from 91 nanometers to 108 nanometers with a narrow polydispersity. Could I have the next slide? And they were injected into animals. And so this is what happens when you inject them into mice. This is unmodified liposomes of the same composition. And this is all of the polymer modified liposomes. And you can see the half-life in hours. For PEG, half-life was 34 hours. Unmodified was 12 hours. So all of the polymers improved the half-life of the liposome. However, two of the polymers, PEG and the PMOX, on the second dose were rapidly eliminated. So this is dose 1. This is 7 days later on the right-hand side. Dose 2. All of these polymers had similar half-lives, as did the non-modified liposome, but PMOX and pegylated liposomes were rapidly eliminated. Next slide, please. And this led to the liposomes accumulating in the liver and the spleen at a higher level on the second dose than they did in the first dose, whereas the other polymers showed no difference on the second dose. Could I have the next slide, please? And when Paul looked into this, he could find that in the pre-immune mice, there was no IgM antibodies against the polymer. In the post-injected mice, there were high antibodies, IgM antibodies, in the serum after the polymers. Next slide, please. There was no cross-initiation observed. That is, if we injected the mice with a PEG modified liposome on day 1, and 7 days later it came back with a PMOX modified liposome and injection 2, the PMOX liposome wasn't at all affected. So it turns out that the antibodies that are formed against the polymer on the liposome surface are polymer specific. Could I have the next slide, please? And the most interesting thing about this study is that it turns out there was a high correlation of the circulatory half-life of the modified liposomes with the intrinsic viscosity of the polymer. So polymers like PEG and PMOX, which had a very long circulation half-life, are the ones that had a very high viscosity. So the circulation half-life is on the left-hand side. The viscosity is on the x-axis. The top bar is for studies done in rats, and the bottom bar is for studies done in mice. So it was a correlation that is species independent, and we were quite amazed by the quality of the correlation, 0.92 and 0.84, and we're pleased with that because that allows you to make some hypothesis about how this is acting. And our thoughts on this is that the polymers themselves are interacting with B cells in circulation, and the viscosity of the polymer retains the liposome interaction with the B cells for a longer period of time if it's a more viscous polymer than it does with polymers that have lower viscosities. Could I have the next slide, please? Okay. And for the initial studies, all of the polymers had the same molecular weight, but PEG is a much smaller monomer unit. So the polymer length for a 2K PEG is much longer than the polymer length of the HPMA, PVP, and the other polymers. And the same is true of PMOX. So what Paul did is went back and he resynthesized a few of these polymers, PVP and HPMA, with a higher molecular weight so that the monomer unit would be exactly the same as found in the 2000 molecular weight PEG. And making the PVP and HPMA polymers longer did not induce a accelerated elimination phenomena. If you can look here, the first dose, 23 hours, coming back a week later, it was 24 hours. And for HPMA, the first dose was 19 hours, and coming back a week later, it was 18 hours. So our hypothesis that we currently hold is that the viscosity of the polymer is the important thing. Its conformation on the liposome surface may vary, brush versus a mushroom. That could also be important. And so it may mean that if you could get the right liposome grafting densities, you could drive the circulatory half-life of these other polymer modified liposomes up quite to long periods. Could I have the next slide, please? So let me sort of turn from that topic to the second area that we've been interested in for some time, along with many other laboratories, is the tumor penetration area. Could I have the next slide, please? So our approach to this was to encapsulate methyl umbelliferone phosphate into a liposome. And so this is a prodrug of 5-MU. And the concept was that it was in the liposome, it would be stabilized. When it was released from the liposome, it would be attacked by serum phosphatases or cell surface phosphatases, would generate the methyl umbelliferone, which is, you can consider it as a prodrug. Can I have the next slide, please? And so we had hoped that based upon work out of Halozyme, that if you they took a pegylated hyaluronidase, injected it into patients, and they could deplete hyaluronic acid in the tumor microenvironment in certain patients. And then they could administer drugs, and they got reported better therapeutic effects. The idea being that you eliminate the HA and you are better able to percolate through the tumor, and you'd get higher tumor penetration. This is a simple concept. It may not be totally correct, and I'll get into that in a second. So that's what we're attempting to do, though. Could I have the next slide? And the idea is that methyl umbelliferone serves as a substrate for the glucuronic acid from UDP glucuronate, and it becomes glucuronidated. And that blocks the synthesis of hyaluronan by HA synthesis in the cell. And so rather than degrading hyaluronic acid, we're attempting to block its synthesis. Next slide, please. And these are the results. Indeed, the HA comes out of the liposome. When it's out of the liposome, the phosphate regenerates into the methyl umbelliferone very rapidly. And it also inhibits the production of hyaluronic acid in the tumor. I won't show you that data, but this was the scheme that we came up with. The animals were tumored on day 0, and then on day 4, 8, and 11, they received a liposome that had methylumboliferone phosphate in it. On day 14, they received the combination of the Doxyl liposome plus MUP liposome, and the MUP was dosed at 18 mg per kg, and Doxyl was dosed at 6 mg per kg, and these are the results. Tumor volume, untreated, treated at the very bottom curve, treat it with Doxyl, and then treat it with Doxyl with the pre-dosing of MUP. You can see in the upper right hand that the untreated animals, most of the liposomes were near, this is an arteriole, fewer liposomes were dispersed throughout the tumor. If you look carefully, you can see some of them in this image. After the 4-dose treatment with liposome MUP, you can see that the liposomes had entered the tumor further away from the endothelial cells in the arteriole. And in fact, if you look throughout the tumor, there are more liposomes in the tumor than in the untreated animals. And that led to both a slowing of the growth rate of the tumor and also a length in the survival time of animals treated with this dose. Can I have the next slide, please? So, what do we expect from an ideal nanomedicine? Well, long circulation is important, the appropriate drug release. I would say that we're not there yet. Navigating past matrix components in solid tumors, diffusion, convection, and action at a distance. We're not completely there yet. Targeting to accessible tumor cells, if they're accessible, we're doing much better. Easy to manufacture. They're reasonably easy to manufacture. There's processes in place that can give you reproducible liposomes, but it is a process. Let me, in the last 5 minutes, turn to this easy to manufacture bit. Before going though, hold it if you, yeah. Next slide, please. This first section of work was done in my laboratory at the University of California. The key people in this were Didier Colli, Paul Kerstead, Saul Himami, and it was done in collaboration with Jean Frichet at UC Berkeley, Cameron Lee, Derek Vanderpoel, Heidi Keller-Ferguson, and of course, Paul. And we had collaborators at the San Francisco General Hospital, Walt Finkbeiner. Can I have the next slide, please? I'd like to turn then to what I would call the company side of my existence. And we became interested in the fact that 80% of new molecules are poorly water soluble, which makes them incredibly hard to formulate. And for compounds intended to be given IV, it's even a bigger challenge. Could I have the next slide? Here are some examples. Itraconazole, desferasirox, nilotinib. These compounds, if you can look at their structure, you can understand why they have low water solubility. Ring structures, rigid, few ionizable groups. Next slide, please. However, they make up a huge market. And it turns out that many of these compounds are orally available, even though they're sparingly water soluble. You can formulate them in an amorphous state, and they can get very high solubilities. It's like, so that is a useful property. Could I have the next slide? The delivery options for these are micelles, cyclodextrins, which are very popular, liposomes by putting the drug in the bilayer, and liposomes with the drug encapsulated. Next slide, please. So we return to this old liposome remote loading technique. And this was a concept was introduced by Dave Deemer back in the 70s. It was picked up on by Pieter in the 80s, who developed a process for remote loading liposomes with a pH gradient. And it was picked up by Hezi Barinholtz while he was at Sequus on a sabbatical, using the ammonium sulfate as the gradient. The idea is you have a drug, it is in a neutral form, it crosses the bilayer on the inside. In this case, it picks up a proton and it forms a precipitate with sulfate. And that gives you that bean-like structure on the inside. Can I have the next slide? It's a very, very powerful technique, this remote loading. But the problem is if you have a crystalline drug that you can't solubilize, you can't load that drug. And you can't get it at a high enough concentration in the aqueous phase in order to load it. And so there's a couple ways that we can get around this problem. One is we solubilize the drug in DMSO or some other solvent. We dilute it in with the liposome. The drug forms an amorphous aggregate and it remote loads. With the crystalline drug, you just add that drug to the liposome and it doesn't remote load. Here's an example. So this is the loading efficiency with the crystalline drug added to liposomes, and let sit for 120 minutes. Absolutely no remote loading. You have drug crystals and you have liposomes, and never the twain shall meet. If you add the liposome in DMSO for 30 minutes, it quickly loads. If you add the drug slowly in DMSO, it also quickly loads. So you have a system that if the drug can form an amorphous solid, you can load it into the liposome. Can I have the next slide? Let me show you this. There's a video here. There we go. Okay, if you hit that video, here's an example. The liposomes here are in a buffered solution. The drug is carfilzomib. It's being added from a DMSO solution. You see when it's added, the drug precipitates. The precipitates are greater than 200 nanometers. They can be filtered out. You then take that mixture, put it into a heated water bath. You can see it's cloudy. And then within a few minutes, boom, the liposome takes the drug up. What happens? You go from a amorphous solid at 200 nanometers, 300 nanometers, and it becomes an amorphous solid inside the liposome with a diameter of about 50 nanometers. When you see this in action with a lot of different drugs, it really is quite impressive. Could I have the next slide, please? You need a trapping agent. If the trapping agent is sodium sulfate, it doesn't load. Ammonium sulfate, you have a gradient, it loads. Next slide, please. If you remove the DMSO from the precipitate by solvent exchanges, so you do it by a 10-fold solvent exchange, or you wash it extensively, or you precipitate and reprecipitate, the drug still loads as long as it's in the amorphous form. Next slide, please. And this is what it looks like. This is liposomal carfilzomib on the right-hand side. It forms a very nice nanoprecipitate inside the liposome. On the left-hand side is a deferoxamine liposome that we were also working on at the company. So this is the carfilzomib liposome. Next slide. This is the solid of carfilzomib, the X-ray scattering. It's crystalline. This is the precipitate of the carfilzomib from DMSO. It's now, you can see it's an amorphous. You've lost the sharp scattering peaks. If you form, in this case, it's a sucrose octasulfate complex. It also is an amorphous complex. And these are the liposomes that have loaded the carfilzomib. And you can see here again, it's an amorphous complex inside the liposome, but it's a very small precipitate. Next slide. Whereas the commercial product, Kyprolis sells about $1 billion of drug a year. It's in a captisol, which is a cyclodextrin, I want to say polymer, it's in a cyclodextrin. And it forms a encapsulated compound in the cavity. The problem is that you need a 5 to 6-fold excess of the captisol to solubilize the drug. And as soon as it's administered into a patient, it dissociates. So the drug rapidly dissociates from the carrier, and it's rapidly eliminated. Next slide, please. Using the properties of, next slide, please. Yeah, here we go. Carfilzomib has a time-dependent effect on cell death. Short time, the IC50 is high. Longer time, it decreases by 10 to 50-fold, depending upon whether it's solid tumor cells or hematological tumors. Next slide, please. So you can see where I'm going with this. Increased exposure time improves anti-tumor activity. Next slide. And what was done at Zone 1 Pharma is they explored a very large number of formulations. On the left-hand side, in the red squares, you can see the elimination curve of Kyprolis, that is the carfilzomib in the cyclodextrin. Its half-life is less than 5 minutes. And over the course of looking at 40 to 50 different formulations, the company was able to improve the half-life of the formulation by a 14-fold, and that led to a 2,000-fold increase in the area under the curve. Next slide. And looking at 2 mouse models, we could use a surrogate marker vehicle. There's no effect on this marker, which is blockage of a protease. The free drug knocks it down and keeps it down for about 6 hours. The liposomal drug knocks it down and keeps it down for, well, longer than 6 hours. And in the solid tumor model, the free drug knocks it down by 40%, whereas the liposomal drug knocks this surrogate marker down almost 95%, and it stays down. So based upon this, we thought it would be a good compound to try to treat these tumors. Next slide. And this is in a murine lymphoma model, and the animals were dosed weekly with either the Kyprolis, the drug in captisol, at 5 mg per kg, or at the equitoxic dose, which is 25 mg per kg. And here you can see at the equitoxic dose, the liposomal drug did a better job at suppressing tumor growth. Next slide. We went to a solid tumor, that is the mouse colon tumor, CT26. And this was a single IV treatment on day 0. And we're comparing carfilzomib in a liposome to carfilzomib as Kyprolis. And also we looked at a combination of vinca alkaloid with carfilzomib. And this was done at putting the compounds in the same liposome with the proper ratio. Next slide. And the nice thing about this drug loading technique is, regardless of the input drug to lipid ratio, and this is 500 grams of drug per mole of lipid, you get 100% loading. And you could co-load the 2 of these drugs to a very high level in the same liposome. In fact, in some cases, we went up to 1.9 grams of drug per mole of lipid, which is really quite amazing. Next slide, please. And when we treated the animals, this is what we observed. On the left-hand side, the black curve is the vehicle. The curve after that is carfilzomib at the maximum tolerated dose. carfilzomib dosed with the vinca alkaloid at the appropriate ratio. The next curve is the liposome carfilzomib. And finally, the curve in blue is the co-encapsulated liposome carfilzomib and the vinca alkaloid. And here again, you can see that the liposomal form at an equal toxic dose is superior than the carfilzomib or the carfilzomib with the vinca alkaloid. And the survival curves are shown on the right. Okay, so this is an example where liposomes can do better. You have dose-independent pharmacokinetics. Could you turn back to that summary slide there? Dose-independent pharmacokinetics. This is the carfilzomib at 25. This is at 5. This is compared to the free drug. So the MTD increases 5-fold in the liposome, the area under the curve 2,000-fold, and the animals are really spared the effects of the drug, which you can see in the lower curve. Could I have the next slide, please? I just want to finish on one small thing. You can take these liposome encapsulated sparingly soluble drugs, you can give them orally. Next slide. And this, in this case, this drug is nilotinib. This is the crystalline form, this is the amorphous precipitate, and this is the encapsulated nilotinib in the liposome. Next slide, please. The animals were dosed at 25 mg per kg, and the comparison is putting nilotinib in hydroxypropyl methylcellulose. That's the formulation that was published by the owner of this drug. And here we make 2 different liposomes, citrate loading in hydrogenated soy PC or an egg PC, and you're looking at blood levels at 1 hour and 4 hours post-dosing. And the liposomes do as well or better than the hydroxypropyl methylcellulose. Next slide. So the idea is that the drugs are placed into the stomach, into the intestine, and what happens is that biosalts disrupt the liposome and release this nanoprecipitate, which then it's in the amorphous form. It can interact with the enterocytes, and it can cross the gut and get into the body. So lipid bilayer protects the amorphous precipitate in the stomach. Biosalts permealize liposome and small intestine, and we think the nanosized amorphous precipitate is then absorbed. Next slide, please. Okay, so this is enough. This technology expands formulation possibilities. So you can get all of these drugs that have solubilities that are less than 2 mg per mil, if they can be remote loaded. You can use them with novel new chemical entities for delivery. You can repurpose drugs, and presumably you can get patent life extension. Next slide, please. So the ideal nanomedicine, is it a liposome? Well, they have long circulation. In some cases, they have appropriate drug release. We still need to do much better here. I would say they're easy to manufacture now. They can be made by microfluidics and by extrusion. They're not yet ready to navigate past matrix components in solid tumors. Diffusion, convection are a problem. And so if you have something that acts at a distance, like a radionuclide that can penetrate tumors for a certain distance, like rhenium, they can be effective. Targeting to accessible tumor cells is certainly readily available now. But I think what I am leaving you with is that there's ample opportunity for researchers to improve nano-drug therapy and improve liposomes. So on the next slide. Thanks so much. And I'd like to acknowledge my students again, Heidi and Aditya, the people at Onyx that we worked with on the carfilzomib, Gertstorm, Alberto Gabazon for the use of 2 of their slides, and the Zone 1 pharma team, Charles Noble, Mark Hayes, and David Tran, without whose ingenious work the remote loading of the sparingly soluble drugs wouldn't have been completed. And thank you.
The first is Miguel Pereira, and Miguel asks, I would like to ask your opinion on cell membrane nanovesicles, aka as cell membrane derived biomimetic liposomes. A promising reality? Question mark. And what do you think of exosomes when compared to these vesicles and liposomes in all aspects? Thanks very much, and regards from Portugal. And he began saying, great presentation and interesting. So Frank, over to you.
Yeah, no, I think the exosome area and reconstituted exosomes are a very interesting approach to this. I think there will be problems in navigating the approval process for these because they're really a much more complex mixture than the lipids and polymers that are currently used in liposomes. In the exosomes, or reconstituted exosomes, you have membrane proteins and oftentimes a number of membrane proteins. Depending upon the source of this material, as I understand it, it can be quite difficult to get reproducible sources of material. And I suspect that that startup companies and companies working in this area are working hard to improve that. But I see a tough regulatory time for this. And I also think there will be difficulties in scaling these up unless the number of components can be limited and these components can be made in a way that provides a consistent source of material. What is the big thing now that is a barrier to complex systems getting approval is analytical techniques have become exquisitely good. And the FDA and other agencies are ratcheting down on what's in the bottle and what is the reproducibility of your formulation. So to my mind, I think these problems can be solved, but I think they're really hard problems compared to those of us that have worked with 1, 2, and 3 component liposomes. Even the liposomes that Pieter has really championed and has developed and put forward, those formulations, the 4 component formulations for the mRNA delivery, I mean, they have 4 components, and there can be some challenges in doing analysis on these. But clearly, these challenges have been solved by money and need and ingenious mentoring of people and companies along the way by those who have developed these.
Thank you, Frank. We have 2 questions from Nitish Kunte, and I'll ask Marshall to read them out to you, Frank.
Here we go. First question. When nanoparticles accumulate in tumors through leaky vasculature, will it be possible for them to return into blood circulation due to higher blood perfusion? That's the first question.
Mm-hmm. Yeah, no, that's a good question. And so, yeah, some of the nanocarriers can egress from the tumor. But what happens even more readily is when the drug is released, it gets washed out of the tumor. There's been a huge effort to get localized targeted release, either by a temperature differential, that is, you make a temperature sensitive liposome. There was a temperature sensitive Doxyl liposome that went into the clinic. They would heat the area of the tumor by various methods. And they really got a very modest increase in activity. And the reason for that is blood flow, you heat it, blood flow increased, permeability increased. And so when the drug was released from the liposome, it was washed out very quickly. And that's one of the challenges for localized release. If you get the drug out of the liposome, it then becomes a candidate for flushing away by blood flow or interstitial transport before the drug can reach the target cell. And so that's a challenge.
The second part of the question, Marshall.
Yep, the second question. If nanoparticle surface, if a nanoparticle surface is modified with PEG or other polymers to prevent clearance by the macrophage system, what percent of nanic, it may be intended nanoparticles, but it reads nanic particles accumulate within targeted tumor cells, and what percentage is engulfed by tumor-associated macrophages?
Yeah, that's a great question. That's a great question. It has been looked at by a number of groups. Dimitri looked at this back in the early 90s with the Doxyl liposome, and quite a few liposomes are actually in macrophages. And so the hypothesis is that the drug is able to escape the liposome in the macrophages and leaks out of the macrophage to kills the tumor. I can't give you the exact number or percentage of liposomes that are in macrophages in tumors. Other people have also looked at this. It's clear that liposomes get into tumors. Some people have hypothesized that that may be actually a very important route of getting liposome encapsulated drugs dispersed through the tumor. That's a hard question to answer. I know that Terry Allen has looked at this a long time ago and looked at where liposomal drugs might be released, and she looked at Doxyl with one of her students up in Alberta, and it's a very nice paper at looking at the fate of liposome contents in tumors. And I just would like to say that the liposome field in the last 20 years, the nanomedicine field, has just been overwhelmed by a large number of groups from around the world that look at different aspects of liposomes. And I've been overwhelmed by the number of papers that have appeared, and it becomes very difficult to assess what all this information means. A lot of it is rediscovering things that were discovered in the 80s by groups in Holland, groups in Canada, groups in San Francisco and elsewhere on looking at the what I would call the pharmacology of liposome encapsulated drugs. I mean, Andy Janoff at the Liposome Company led a big effort with Eric Mayhew and others to look at what happens to liposome contents. And Andy actually was the first CEO of Celator, which then went on to develop the two-component liposome with a very fabulous group up there. And of course, that group, we're all associated with Pieter at one form or another, either at the Cancer Institute or, as was said earlier, Pieter is like an oak tree. You know, there's a lot of oaks that have fallen out of the tree, and they've, rather than developing oak trees, they've developed liposomes. Anyway, it's a great question. And the one thing that I didn't answer is what percentage of the dose gets to a tumor with liposomes. Actually, depending upon the tumor, it can be up to 7 or 8%. It can be very high. And in comparison to some of the nanomaterials that are smaller, made out of metals, even if they're pegylated, you know, their half-time is 15 minutes, 20 minutes. So they don't have the window to go through the tumors for multiple passes. And so when you read reviews about nanomaterials, and it says, well, we're only getting 0.7% of the drug into the tumor, it's because the particle is eliminated so rapidly. But with liposome, it has many, many passes through that tumor, even if it's not long circulating. And you know, its half-life is an order of magnitude or better than some of these small nanomaterials. And as I mentioned, you can get 7, and I've seen up to 10%. And Carposi's definitely was 10% of the material that was injected ended up in those lesions. And that's why they're the oldest kid on the block.
Frank, I'm going to take a question from Zara Bakari. She's asking, do we see a challenge in terms of ubiquination of liposome delivery systems, and how do we circumvent that?
Well, yeah. I usually think of ubiquination as something that happens with proteins. So I really can't respond to that because I'm just not knowledgeable enough in that area. And I'll have to punt that question to somebody else. But I think where that's coming from is getting back to exosomes. And the question about exosomes, which have proteins, the ubiquination could be a factor in those, as I quickly think about that.
Okay, thanks. There's two more questions I'm going to try to squeeze in, and I know people may have to go, but Sartak Garg has a question. Thank you for your presentation. I'm wondering how important you think particle number is in dosage, and relatedly, whether you think if it's better to raise DL as much as possible to lower dosage volume.
That's another great question in terms of the pharmacodynamics of liposomes. So what I've shown you is that with this sparingly soluble drugs, we can really make highly encapsulated drug levels. And so the particle number goes way down. We haven't done those experiments. We haven't looked at the exact same dose, but at different particle number. And those would be great experiments to do. To answer that question, and I'm afraid the answer is going to be confounded by the drug that you select. If the drug can escape its final fate, that is, come back in circulation, it might not make a difference because the local release may not be the factor that's killing the tumor, but this very slow metronomic type dosing that may come out of liposomes, that may be the factor that's working. And so for those types of compounds, it could be that particle number, that is the drug to lipid ratio, doesn't make any difference. But for other compounds that may be rapidly eliminated once outside the liposome by metabolism or degradation, like carfilzomib, having a higher drug to lipid ratio could help you. In that it would concentrate drug at the site and potentially overwhelm inactivation processes, the epoxide or other compounds that are degrading the drug. But that really is a fascinating question, and that it's something that I don't want to sound too pejorative, but I think it's important to work on new compounds. I see lots of publications still working on doxorubicin. And doxorubicin is a story that is fairly well worked out. And my advice to any young students and investigators in the audience is pick up and study some promising new sparingly soluble compound, because that could allow you to look at some new phenomena that are understudied in the nanomedicine area. And, yeah, that's sort of preaching.
Great advice. Thank you very much. And final question, Anna Alexieva asks, please explain about the process of loading the drug from solution into DMSO. What are the driving forces of this process? So that might be a very detailed question, but maybe there's a shorter answer to that before we.
Yeah, no, it really is a short answer, and it really is a fascinating answer from my point of view. When you load a drug into a liposome, you have a huge membrane surface area. So if you have 10 micromolar per mil liposome concentration, huge surface area, it's bigger than the size of a basketball. And what you're doing is you're taking that amorphous drug and it's banging against the surface of the liposome. Some fraction of that drug leaves the amorphous complex, goes into the membrane, and crosses the membrane. So when you think about it, it is exactly like taking an amorphous drug, putting it in hydroxypropyl methylcellulose or cyclodextrin, and putting it into the stomach of a person or an animal. You have this huge surface area in the intestine, and the amorphous drug bangs into that membrane. Some of it goes across, and it's absorbed. There's a sink there. The same thing happens in this amorphous loading process that Mark and Charles and I invented. And so huge surface area, driving force that once the drug is in, it can't come out. And that, we think, is the mechanism behind this. And it doesn't make any difference if you put the drug in DMSO and it forms an amorphous precipitate in the presence of the liposome, as I showed, or you pre-make the amorphous precipitate, just buy an amorphous complex, throw it on the liposome, and boom, it is soaked up like a sponge. It really is quite fascinating. And you know, when you think you've seen everything and you see this, you go, oh, wow, that's really neat.
Well, Dr. Szoka, this has been a really neat lecture. Thank you so much for delivering the April 2022 Pieter Cullis Invitational Lecture. And today's participants, thank you for joining us.